So we're going to be talking about photosynthesis, and I'm going to divide the content for this week into 2 mini lectures. So in this first mini lecture, I'm going to start with an overview of photosynthesis, then describe the experimental evidence that led scientists to discover that there were 2 linked pathways. One of them required light and converted that light energy into chemical energy. And the second pathway uses that chemical energy to make sugar. We're going to talk then about what chloroplasts look like. You saw these structures in Eladia last week. And we're going to talk about pigments in those chloroplasts and how they absorb light. And some of this is going to be review from your week 1 lab. We'll then start to talk a little bit more about how chlorophyll absorbs light and how that light can be converted into useful forms of energy. So photosynthesis is a really cool process because it converts light into chemical energy. And this supports basically every food web on the planet. And so let's take a look at what happens during photosynthesis. So we have here carbon dioxide, and that carbon dioxide, that gas of carbon dioxide, is in the process of photosynthesis is converted into sugar. So that's shown here. And water is converted into oxygen gas. So let's take a look at this carbon dioxide and see what happens to these bonds in the process of photosynthesis. So carbon forms a polar covalent bond with oxygen, right? Oxygen is more electronegative than carbon. And so it unevenly shares these electrons, right? So carbon in this example, it doesn't have as much of the electron pull as oxygen. But when it's converted into sugar, the carbon is now bonded to other carbons as well as to hydrogen. And in those bonds, those are non-polar covalent bonds, the electrons are shared evenly. So because carbon essentially gained electrons, even though it wasn't a full transfer, we say that carbon is reduced, right? Because the electrons are pulled to carbon, so carbon is reduced. And this process of reducing carbon, it's going to take energy because here carbon dioxide has less potential energy because these bonds are much more stable. The polar covalent bonds are much more stable than non-polar covalent bonds, which means that they take some sort of heat or energy to break. In the case of water, we go from having polar covalent bonds to having non-polar covalent bonds. So in this case, the electrons are pulled away from oxygen and oxygen is oxidized. So these are partnered together, these redox reactions. And this process occurs in multiple steps, and we're going to talk about those. But first, I want to begin by talking about this evidence that there are 2 pathways, okay? So here's that formula rewritten where we have carbon dioxide, water, and light. And that gets converted into sugar. So this is my formula for carbohydrates, CH2O, and oxygen. And originally, scientists hypothesized that this carbon dioxide, right, was combined with water in a single reaction, and then that oxygen was released in carbon dioxide was released as oxygen gas. But then Cornelius van Niel performed these experiments in these purple sulfur bacteria that led to some new insights. So in this reaction, we have carbon dioxide, and H2S takes the place of H2O. Okay, so that's what that looks like here. And so this is going to allow us to follow what happens to this oxygen in water, right? And the hypothesis is that this is going to get combined into the sugar. Okay, and so what they found was that you ended up with sugar, H2O, and 2S. Okay, so now this oxygen here, right, is now sulfur. So if we're following this, right, this means that the oxygen in the case of here, right, this would be H2O, right? This doesn't come out in the sugar. The water comes out in what would be the oxygen gas, okay? So this showed that the H2S and carbon dioxide, they didn't combine directly to make the sugar. And that the oxygen in CO2 is not released as oxygen gas. Okay, so this showed that there were two separate reactions. And these experiments were later confirmed by radiolabeling. So they showed by radiolabeling carbon that carbon in CO2 was incorporated into sugar. So the air that we exhale will get incorporated into the sugar that plants make and will become part of the mass of plants. And the oxygen, right, so when they radiolabeled oxygen in water, that came out as oxygen gas. And so there were two separate pathways. And so here's what we know today. So sunlight is captured in these light capturing reactions where water is consumed and it is released as oxygen gas. This light capturing reaction, where we convert light energy into chemical energy, leads to the production of ATP and NADPH. NADPH is basically a phosphorylated version of NADH. And that chemical energy that's generated in this light capturing reaction is used to fuel the fixation of carbon dioxide. So we're going to consume carbon and make sugar. So convert it back to a useful form of chemical energy. And we'll talk more about these two pathways in the next video. But first I want to talk about how chloroplasts and how plants are able to capture light. So where does photosynthesis take place? It takes place in the chloroplasts in plant cells. Okay, so here's our plant cell. We looked at these. These look a lot like the Elodea that we looked at. Maybe the Elodea were a little bit more stretched out. And we have all these chloroplasts. Of course, there's other things in the cell like mitochondria and nuclei. But we're going to zoom in on the chloroplasts, which are really easy to see. And the chloroplasts have an outer membrane as well as an inner membrane. And then they have these structures on the inside, okay? And so I'm going to break these apart in a little cartoon. So here's our chloroplasts. And inside the chloroplasts, we have these flattened structures that we call thylakoids. So this is our flattened structure. And the inside of a thylakoid, we refer to that as the lumen. And these thylakoids, these flattened stacks, they stack up against each other, and we call a stack of these thylakoids granum. And then outside of the thylakoid membranes, we call that the stroma. And so these are where our reactions are going to take place today. So it's useful to have this diagram handy so you can refer back to it and see the location that we're talking about in the cell. So recall from the first lab that photons of light can be absorbed by a pigment, transmitted, or reflected, and that the pigments, as we saw, only observe certain wavelengths of light. So to understand how pigments capture or absorb light, it's useful to review the properties of light. So light is a type of electromagnetic radiation, and that is a form of energy. And physicists describe light as both wave-like and particle-like. So like sound waves, light can be characterized by its wavelength. And that's the distance between these two crests. And visible light is the wavelength that humans can see. So here's the entire electromagnetic spectrum, and humans can see this visible wavelength of light. And this wavelength of light, or some of these wavelengths of light, are absorbed by chlorophyll pigments. And these shorter wavelengths of light, meaning that the distance between the two peaks is shorter, has higher energy. And these longer wavelengths of light, with a greater distance between the wavelengths, has less energy. So this means that blue light has more energy than red light. Okay, so that's some of the properties of light, being that light acts wavelength. To emphasize the particle nature of light, physicists also point out that light exists as discrete packets called photons. And pigments can absorb some of the energy from photons, but how? Okay, so let's take a closer look at chlorophyll and determine what parts are important for this activity. So the tail is made up of an isoprenoid, and this binds to proteins in the thylakoid membrane, which is a lipid membrane. And the head has a magnesium ion, and this is the part of this lipid-soluble molecule that can absorb the energy from light. Also, a small shout out for carotenoids. These function as accessory pigments. So they can extend the wavelength of light that's absorbed by plants or other photosynthetic organisms. But they also protect plants from ultraviolet light, or the ultraviolet part of the electromagnetic spectrum. And this light is strong enough that it can knock electrons off of atoms. And this creates harmful free radicals. And so when this happens, the carotenoids can accept those free electrons and quench the radicals. And as a matter of fact, often carotenoids are targeted, their production is targeted in herbicides. And once these are eliminated, the plant will die because they don't have that protective function. So they play a really important role, even though our focus for today is going to be on chlorophyll. So what do we mean when we say that chlorophyll absorbs red or blue light? So when a photon strikes the head of that chlorophyll molecule, the light energy is going to be transferred to chemical bonds. And in turn, an electron becomes excited. And so this electron is bumped to a higher energy state. And these excited electron states, they're discrete and not continuous. And this can be represented by this energy scale. So these levels are properties of an electron configuration in a particular pigment. And so at this 0 energy scale here, this is the ground state of our electron. And for example, when red light strikes our pigment, this can bump an electron up to a higher energy state at level 1. But when blue light, or photons from blue light, strike the pigment molecule, this can bump electrons up 2 energy levels. But because green light or green photons are intermediate, they're not absorbed by the pigment molecule. And so those electrons can't be bumped up to a higher energy level. They can't be excited. So once, let's say we've put blue light on our chloroplasts, what happens to the energy from those excited electrons? Well, it doesn't last very long. So on the level of picoseconds. So what happens to that energy that was absorbed? So in an isolated solution, as you saw in lab, it can drop back down to a low energy state, emitting light, and that light we call fluorescence. Alternatively, it can drop back down to a low energy state and release heat. So it can just turn back, right? And this is typically what happens in a solution of chloroplasts, or a chlorophyll solution. But in chloroplasts, 98% of the electrons drive processes related to photosynthesis. And these electrons are going to emit resonance or participate in a redox reaction. So let's take a minute to explain what the difference is and what these are. So in chloroplasts, 200 to 300 chlorophyll molecules work together in this group of that's called a photosystem. And there are 2 photosystems. We'll talk about them next. But they consist of accessory pigments, like the carotenoids, antenna pigments, so those are depicted here as these light green structures, and reaction centers. And these are all organized in a precise way by accessory proteins. So when light strikes an antenna pigment, the light energy is absorbed and you get an excited electron, but it transfers not the electron, but energy to the next accessory pigment. And that accessory pigment, its electron will become excited and fall back down. And the energy, when it falls back down, is transferred to another accessory pigment and another one, directing that energy towards the reaction center. And we call this transfer of energy, but not the electron, from chlorophyll to chlorophyll in these antenna pigments, we call this resonance energy transfer. And as I mentioned, proteins organize and tune the absorption potential of these antenna pigments to direct the energy to this reaction center. And so once the energy reaches the reaction center, there's a special pair of chlorophyll molecules in this reaction center, and we call them the special pair. And the energy transferred here will excite these electrons. So we have these electrons being excited, but instead of energy transferring away like it happened in resonance energy transfer, the electrons are actually transferred to an electron acceptor in a new compound. And this is where the next lecture is going to take over. So what happens once you transfer this electron to an electron acceptor? So in summary, photosynthesis converts light energy to chemical energy via pigments. Distinct wavelengths of light are absorbed by pigments, and the light capturing reactions or the light capturing pigments are organized into photosystems. The antenna pigments in those photosystems transfer energy from excited electrons towards the reaction center via resonance, and pigments in the reaction center transfer their electrons to a new compound, which will take us through photosynthesis. So that's it for now, and I'll see you in the next one.